Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, Riffen, I'd like to welcome you to Will COVID-19 Produce a New Normal in R&D? Faster, more decentralized, more digitized. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I'd like to introduce our moderator and the first speaker for today. He is Dr. Timothy Gardner, PhD, founder and CEO of Riffen. Welcome, Dr. Gardner. The presenter ball is yours.
Thank you, Elizabeth. Let me pop my screen on. There we go. So thank you for joining us this morning. As Elizabeth introduced, we are here to talk about, to ask the question if and how COVID-19 and other forces are driving biopharmaceutical R&D and manufacturing development cycles towards more decentralized R&D activities. And if so, the further question of how companies are adjusting to this seemingly competing needs of faster and more decentralized. The event today is sponsored by Riffen, and I'm the CEO of Riffen and founder, and I'll be starting out with a presentation shortly after these introductions. I'll be talking about how Riffen has approached the challenge of delivering products to market twice as fast as historical averages. And after me, Anastasia Christianson, who's the VP of R&D Business Technology at Janssen Pharmaceuticals, will be giving a presentation on digital transformation in pharmaceutical R&D. And her talk will be followed by Oliver Hesse, who is the head of biotech data science and digitization at Bayer Pharmaceuticals. He'll be speaking on the digital continuum from development to manufacturing. We'll then wrap up with a brief panel discussion where I'll be hosting Oliver and Anastasia to follow up on both questions from the audience as well as a few questions that I've prepared in advance. So I'm really looking forward to the event today, and again, thank you for joining. Don't forget to send your questions to the chat room. So to begin, I would like to kick us off with a perspective on the challenge of faster R&D, how to get to market more quickly. I'd like to start with the story of the Vasa. So this is a ship many of you probably visited in Sweden that was built in 1628 by the King of Sweden to intimidate the Poles when they were at war. And his idea was that if he built a ship twice as high as any other ship in the world, they would be so frightened by all the cannons and the height that they would essentially back down in whatever they were fighting about at the time. So they built this ship. They didn't have any calculations or plans. They just added 2 decks to the normal ships that they built. They launched it off with 200 dignitaries. It went 200 meters out into the harbor. A gust of wind came along, blew it over, and it sank to the bottom immediately. So I laugh at that. But I also, when I hear that story, it always reminds me a little bit of pharmaceutical development. Because so much of how we develop drugs today, despite all the incredible minds and advanced technologies, is still a bit random. It's a bit of a guess. And we've even built that into the pipeline with how we funnel targets down to prospective drugs and prospective drugs to realized drugs. Some years ago, this was about a 1 in 25 chance that a specific validated target would make it to a drugged and marketed product. It's actually gotten worse since then. In the past 10 years, it's a little worse than that, about 10% worse. And the cost of development has just continued to accelerate, increase. This is the averaged or amortized cost of all the drugs that are successfully produced as well as all of the failures. A successful drug might be less than that by itself, but the industry as a whole is investing almost $2.6 billion per successful drug these days. That's something many of us are very acutely aware of and have been for a very long time. So the trouble with this, or the approach in some sense, is in my mind like the Vasa. We basically, instead of trying to, well, we certainly try to get smarter, but nonetheless, we tend to approach this problem by just launching not one Vasa, put 25 more VASAs out. We try over and over to do the same thing in the hopes that if we just do it enough times, one of those ships is going to make it. But obviously that leads to a lot of waste, and that's why the cost has been going up. And the truth is we have seen problems like this before. We've seen this in the automobile manufacturing industry and many other manufacturing industries decades and decades ago. And this example shows that we don't have to live with that type of approach. Back in the 50s and 60s, in fact, we had a similar kind of production philosophy. Make your cars, test them at the end with a QC group, and if they didn't work, throw them away and go make another one in hopes that it would work the second time around. That's an enormous amount of waste, perhaps 50% of the industrial capacity going to producing junk. And around the late 50s and 60s, the concepts of total quality began permeating into the Japanese auto industry. It gave us a very nice control experiment, because as they adopted those methodologies, their productivity in the manufacture of cars tripled or even quadrupled in those years. The US auto industry continued as they had with the same practices and saw no gain in productivity. And that's a transformation I believe can take place and is starting to take place, in fact, in the pharmaceutical industry. My basis for this belief is actually industrial biotech. This is a plant in Brotas, Brazil, owned by DSM, that was built by a company called Amyris. And I had the good fortune to work at that company about a decade ago. We were building genetically engineered yeast and scaling them to these reactors, which are 200,000 liter aerobic bioreactors. We actually managed in about a year or a couple of years to improve our processes from the initial state to a operating state where we improve the time to delivery of new technologies from 12 months to 3 months. So we could take a strain from the lab to full-scale production in 3 months, which was 4 times better than where we started. Actually, it's more like 5 times better. We also improved the productivity of these processes while getting these technologies to market much faster. And these experiences led me to the deep belief that that many of the delays in development that we see in pharma and other parts of biotechnology are not inherent. And in fact, we're even seeing that now with the extraordinarily rapid development of drug candidates for COVID, something that gives me incredible hope, not just for the immediate problems of COVID, but also for a dramatic shift in how we approach pharmaceutical development going forward. So why? How do we get things to market faster, to full scale faster? And it really comes down to breaking down the barriers and increasing the understanding of the processes we build. And that's through quality. So this is how we started. Very noisy experimental processes that were not reflective of full-scale conditions. And then what we saw when we transferred our technologies from process development to manufacturing was a huge drop in performance, consistently failing to achieve in manufacturing what we saw in R&D. And then we improved the way we worked, both improving the quality of how we did our research and development to get rid of a lot of the noise, which allowed us to see for the first time the structure of our processes and apply sophisticated process analytical technology with fidelity, but also taking the conditions, the reactor conditions of full scale and making sure they were transparent and shared with the R&D team and vice versa. So breaking down the silos between process development and manufacturing and treating it as a continuum had extraordinary benefits to the acceleration of our performance or our scale up and the right first time scale up. So one of the things that drove this improvements in how we perform is the nature of industrial biotech. It's a low margin business at extraordinarily large scales. So these are just some of the numbers of how intensive these processes were. 700 grams per liter sugar, that's almost solid sugar. 100 grams per liter biomass and 50 grams per liter per day productivity. That's anywhere from 10 to 100 times the intensity that's typically observed in a biopharmaceutical process. you don't have much margin for error, and that drives improvements in how you work. So the question is, what about biopharma? For so many years, it just wasn't necessary to improve the performance because the cost of a drug was so far beyond the cost of its production. You could tolerate a lot of inefficiency in production. What you had to get right was quality. You could not compromise in any way on the quality of a biopharmaceutical product. So that for many years has been a trade-off, but that's changing. The opportunity now is to get both. And the thing that's driving that is the nature of the drugs we make. They're becoming more targeted, more precise to smaller and smaller diseases, and they're becoming more complex in their character. Biologics now take up 40% of active drug development programs in the R&D pipeline, and almost 50% of the drugs approved are now orphans, which by definition target smaller populations with smaller production runs. The result of that is the need to produce many more products because there's fewer or more products targeting fewer patients, and smaller batches. So that drives the need for flexibility, and flexibility in a plant is enhanced by intensification. If you can get the job done quickly and then switch over to the next batch, you can get a lot more done. And we've been seeing this trend for more than a decade with the shift towards flexible single-use systems, increases in the intensification of processes, and higher use of CROs and CDMOs to gain flexibility and performance in production. But the challenge in all of this is that as we get more flexible, as we get more distributed and more dynamic in the way we produce products, we are also limited by some of the old practices of how we worked when you'd make one drug for 20 years in one plant. And the challenge is the transfer of process knowledge between the many teams and sites that contribute to its creation across the development lifecycle and the manufacturing lifecycle. So what's limiting this transfer is some of the manual and siloed practices that we still use, which are very much like the same ones we started with in that industrial biotech context. Fragmented information about the processes we are creating, lengthy manual tech transfer documents and process changeover, site-specific variations in the equipment we're using, and then the result of that is an incredible pain in assembling data to understand how our processes work. The very same problems we experienced 10 years ago in trying to get those yeast up to scale. This is affectionately called data wrangling, but anybody who does it knows this is nothing but pain and frustration. And along the way, I personally realized that I went through a sort of transformation in how I thought about science, and that led to the formation of Riffen, which I'll describe in a moment. But that transformation was the realization that when I arrived to join the R&D team to make a genetically engineered yeast, the product of my work wasn't actually the yeast. In R&D, the product of your work is the process by which you make that yeast. And that's what we were transferring to manufacturing. And so I shifted my thinking and realized that what we really needed to focus on was the articulation of these processes in each stage of development and the smooth, efficient transfer of that knowledge as it grows and matures towards manufacturing as a kind of continuum. And for that, we needed a system that could not only capture process information very clearly and in a shared way, but also something to integrate the data from those processes with the processes that produced them and execute them. And this became the basis of Riffen Nexus, which is the product that Riffen makes, which is a cloud-based data system I'll briefly describe before wrapping up my remarks. So Riffen Nexus is a process data system that provides a life cycle management backbone for design, analysis, and improvement of R&D processes and products. It's focus is to take those process designs and the data you collect and automatically integrate them for real-time analysis of process performance and scientific experiments to address that data wrangling problem that so plagued myself and others. And to do that, we support the entire scientific learning cycle with technology. So it starts with process design, execution, and data capture, integration, and analysis. And it looks like this. This is the interface to Riffen. Your entree to experimentation is actually describing the steps of whatever scientific process you might use, whether that be a fermentation processes or upstream bioprocesses illustrated here, an analytical chemistry method, or an experiment in cell line screening. You capture the flow of materials through the process, the parameterization of every step, its upper and lower spec limits on all of the different parameters you run and targets, procedural information, videos, text, etc. And then you can execute those processes. You basically you take that template we were just showing, you make a copy of it, and then you can put your samples and batches and materials and reagents into the flow, and it all connects up. It links all those things together, those batches and samples, into a genealogical flow so that you can integrate data across unit operations and also across time and experimental variations into one giant data table for analysis. This is all done automatically and in real time, and then you can feed this to any of the best-in-class data analytical, process analytical technologies and tools available on the market, which plug into that flow of information and allow you to compare batches across scales or across experimental variation. That foundation of process analytics and process design is then enhanced with a life cycle approach. So every version of your process that you create is captured as a version with the experiments attached to it, and then you can trace the history of that over time, which leads to the ability to start to do with your scientific and production processes the same thing you do with a Microsoft Word document that you might compare and redline between collaborating people. You can create a diff report. You can do the same thing with source code. What's the difference between this version of the process and that? And see how those changes impact performance. And where that leads ultimately for Riffen and its collaborators and partners and users is a life cycle approach to product development, essentially the realization of that sketch that I showed from 2014, where you can iteratively improve using real-time data your scientific processes, their performance, their quality, and the experimental outcomes from them until you're ready to transfer to manufacturing, and then digitally transfer that process as a blueprint to the manufacturing floor in the very same way that you can transfer a computer-aided design file for a discrete part on an automobile to a manufacturing facility using a CAD/CAM approach. So in essence, we're striving for and starting to realize this CAD/CAM concept in bioprocess. And the payoff has been profound. So for customers and partners who have used Riffen to date, we've helped them achieve pretty dramatic gains in performance, from doubling the pace to market to reducing the effort required to deliver on goals by 50%. And one of those customers who's been so particularly successful and really led the way in harnessing this new approach is Novazymes, which, kind of bringing this full circle to industrial biotechnology, was able to combine Riffen's technology with their own automation systems and data analytics to cut the time to market for their breakthrough biofuels products in half. So they took 4 products to market in 18 months, half the normal development time, and took a significant position in the market that they entered with these products. Does your process work to use some of Van der Waals processing mining techniques? I don't know if I read that correctly, but I'm not actually familiar with the Van der Waals processing and mining techniques. I will say that what we offer is the ability to apply all sorts of advanced machine learning and AI technologies to the data set. In many ways, that was the core concept of Riffen, was to make such techniques possible. And the way it does that is it takes what's normally fragmented data sets and it augments those with many data sets combined across unit operations and across time, so that you can build up through that aggregation of information the kind of scale and depth of data that is necessary and is bread and butter for machine learning. And it also contextualizes that data with controlled vocabularies, so you don't have to clean up what's usually a mess of varied annotations of information. And I'm struggling here with the chat window, which is literally 2 lines big. Let me see. Do you have any insight on how to, this is another question, do you have any insight on how the changes in biopharma research and manufacturing from COVID will affect the industry as a whole? That's a great question. I'm going to pause on that one. I'm going to actually put that in the list to ask at the end of this seminar when we have a panel discussion. And the last question I'll take before turning it over to Anastasia is it possible to integrate Riffen Nexus for strain development R&D? And the answer to that is absolutely. We have built full integration with strain engineering platforms. So Riffen itself doesn't do molecular design of proteins and DNA sequences, but we have integrated with such systems, and Riffen has served as the inventory system for those designs. So the design system deposits the strain information into Riffen, and then Riffen picks that information up and passes it into the experimental and process context, which then allows you to do structure activity relationship and data mining, because all of the information is linked together from DNA and protein design all the way to fermentation, analytical chemistry, downstream processing, and even formulations on those products. Okay, so I'm going to pause there, and I'd like to turn it over to Anastasia for her remarks. And I'll start by introducing her. So Anastasia Christianson is the Vice President of Research and Development Business Technology at Janssen Pharmaceuticals. She has over 20 years experience in biotechnology and pharmaceutical industry, building and leading multidisciplinary informatics and IT teams, driving change and simplification of technologies and processes, and delivering value through innovation. She's worked in discovery and development, managing complex portfolios, translating scientific and medical questions into innovation solutions. And her areas of particular strength include strategy, translational medicine, biomedical and health informatics, evidence-based decision-making, and big data exploitation. So today she's going to speak about digital transformation in pharmaceutical R&D. And I'll be monitoring questions as things go along, and we'll either take them at the end of her remarks, or we'll take them in the panel discussion at the end. So keep sending them.
All right, so the first thing that I need to do is to do the disclaimer for my company. So this presentation is for information purposes only. It is my ideas and examples, of course, informed by the work that I do, no referrals, and I'm not providing any advice or counsel. So let's then get moving. I usually like to start by really setting the stage of like a definition. What is digital transformation? So when I'm talking to you, what am I thinking when I'm thinking digital transformation? And the easiest way to do it, I always start with Wikipedia, and sometimes I have to move on. But in this case, there's a pretty good definition in Wikipedia actually on what digital transformation is. So it's the novel use of digital technology to solve traditional problems, right? Being able to do things better, faster, using technology, and employing new innovation, new ways of doing things, and being creative in supporting problems that have been around for a while, but we now have the capability and the technology to move on them faster and better. It's really the adoption of technology in R&D is not new, but it is definitely being accelerated and has been accelerated. So what are the drivers of digital transformation in R&D? Yeah, I've got on the right is speed, efficiency, quality, and cost. These are the things that have plagued us for years, actually, in trying to do things better, faster, cheaper. But really, the number one focus is on patients and patient outcomes. So can we get better patient outcome? Can we get better results? And can we get to the patients faster? Can we speed up getting the important medicines to patients faster? Faster also it necessitates excellence. So you don't want to get low quality or do low quality work. You must be doing really reaching the next level of R&D operational productivity. Driving scientific and technical innovation, really being at the forefront of using the next best technology and effectively really one of my passions being data-driven decisions. So using technology to drive the generation of the data that will help inform our decisions, and then also using technology to make best use of the data. And we hear a lot about that now with advanced analytics, machine learning, AI, and so on. There's another drive actually to digital transformation, and that's externalization. I've got a cartoon here that shows by yesterday. I really should have put quotations on there. It's not literally yesterday, but a few years back when I first started, certainly in the pharmaceutical industry, we had partnerships, but by and large, we did most of the work internally, or a lot of the work internally. Over the years, and if we look at where we are today, a lot less being done internally, a lot more partnerships externally. What that does is then, it necessitates us being able to be very well connected between ourselves, our CROs, our academic environments, our hospitals, payers, healthcare providers, and so on. And it does get pretty complicated. This next build isn't intended for you to actually read all the details, but it shows a little bit. I have another slide that I sometimes use. It shows a little bit of a spaghetti, right? And there's just way too many connections between different folks. And it's not a one-to-one. The connection isn't one-to-one. It's not just that, if you're sitting in a pharma, you need to have contact with everybody else, but everybody else can sit in their silo. There needs to be interconnections. So effectively, this is the best picture that I could think of, is linking our computers across the world from different partners and with ourselves, actually, to be able to get access to the data faster. Enablers to this are, of course, the large amounts of data that are being generated. I've got a picture here of the Internet of Things. Beyond that, just the exponential growth of data that we're generating. The multitude of, I call them gadgets, of sensor technology and different technologies that can be used not only in the lab, but also in the clinic in order to collect data and collect data sort of in the moment. And also the technology that we have now to enable us to better analyze and visualize the data and look at a multitude of data sets and data types in an integrated fashion. So these are all enablers, so technological enablers for adoption of digital technology in R&D. And then if I look at the labs, if I break it down to the lab and the clinic in a way, if I'm looking at across R&D. So in the lab, digital transformation started, again, many, many years ago with high throughput technologies, high throughput screening, and making compounds in parallel and so on. Moved into the biology arena, initially with genomics, and then proteomics, metabolomics, many, many other omics technologies, and just continuing on digital pathology, robotics, data virtualization, robotic process automation, and so on. So many, many different technologies that are both accelerating what we do, but also accelerating the generation of data that we need to make better use of. When we look on the clinical side, clinical research and clinical trials, again, we've had an increase in smart technologies being leveraged to make measurements in the clinic more real time. And at the same time also enhancing the patient experience, being able to share with them more information in real time or even early before they sign up for trials, and being able to get the drug to the patient or to the sites actually faster, better way. And last but not least, in all of these, improving the efficiency with which we get information. So reducing the amount of paper that we use to collect patient information and collect it more electronically and being able to then access it much more easily. So each one of these has been adopted at varying pace. So I'm not here to give the impression that it's nirvana and everything is all electronic. We are in the process of increasing our digital transformation, and it varies. So if I were to say efficiencies is an area that we spend probably a lot more time and effort in, and the leveraging smart technologies, it's in pockets depending on the therapy area, the disease area, the trial, the site, and so on, what's possible. I'll focus a little bit on sensor technology and remote monitoring because that's important both for understanding the patients better and understanding the disease progression in a patient better. There are many sensors that are currently either in use or being tested. The easiest one for everyone to wrap their head around is actigraphy, right? Fitbit or whatever your favorite step counter is. Many of us have that and do that not because our doctor told us to do it, but we can also use that data in order to better understand what is inhibiting someone from moving or how much they're moving, and for many different disease areas, for example, and sometimes in indirect ways. So, for example, if an RA patient is on a medication and suddenly is not moving as much, is that because they're in more pain, or is it because they're in less pain and can do other things like sitting to knit, for example, or sitting to do something to write, and they wouldn't have been able to do that otherwise if they weren't on the medication. So it measures different things, and we always need to think about coupling that with patient-reported outcomes as well to really understand completely what it is that we're measuring. There are also other sensing motors or sensors that we can use for monitoring motor skills for Parkinson's patients, Alzheimer's patients with voice recognition right now, also looking at trying to understand if the sound, if someone's sound changes when they are approaching, when they are in a depressive state, diabetes, many other applications. And as I mentioned earlier, we're not using sensors completely now for everything, fit for purpose, and also as people become more comfortable with seeing positive data coming through. And the graph all the way on the right is actually quite interesting to look at the gap between the patients and physicians and the adoption of sensor technology variability, if you will. This data is a little dated, but certainly I'll mention a little bit later, the pandemic, I think, has accelerated some of this out of necessity, but certainly there are differences between eagerness of patients sometimes to use technology and the caution of a physician, rightfully or wrongfully. Oftentimes they have very, very good reasons to utilize that, and we need to understand that and move beyond that. So if I look at, very briefly in the slide, if I look at technology adoption, not only in R&D, but because, of course, we're in the healthcare business, understanding what technology adoption is like in the healthcare setting, it's quite informative, actually, and it impacts clinical science. And here are a few examples that I'm sure I can add a lot more, and some recent ones as well, but it gives you an idea of how healthcare is becoming much more of a digital business if it's from the perspective of adoption of electronic health records, willingness for a doctor to have a video conference call with a patient, the use of sensing technology and monitoring of patients remotely, medications actually. There are ways to keep track of medication adherence, and so on. And also, especially now, the interaction with the patient and the speed with which a patient can get advice from a physician or a healthcare practitioner. So this increased technology use fuels generation of large amounts of data, complex data, diverse data sets, the need to look at information access in a different way, because as I mentioned in a few slides ago, the data needs to be accessible by multiple parties, and you need to have some governance around that to make sure that patient information is following HIPAA rules and et cetera. Need to access information globally because we are a global organization and we are working with partners all over the world. You need to be able to trace where the data has been, who has used it, need to access data almost instantaneously if you want to understand a patient's response to a drug, for example. Really, the faster you can get to real data, the faster and better you can make a decision that can impact the next phase of a patient's journey. So, enter global pandemic. With all of this that has been already going on to speed up the adoption of digital technology in research, in clinical, what happens when we have a global pandemic? Broke it down into two parts. In the lab, maybe starting there, probably the most difficult area because you need to run experiments, but also thankfully because we had been going through a digital transformation, facilitating, initiating, and monitoring experiments remotely. So while our scientists might need to go in to start an experiment or replenish reagents, they have been able to start and stop experiments. During the pandemic, actually, someone developed an approach for a robot to be able to move around the lab as long as there are no doors, because the robot can't open the doors, but move around a lab and monitored by having a camera that the scientists can actually look at to see if something has aborted. Is there a jam somewhere? Is there something? What action should they take? Because they can actually see the instrument and take appropriate action. With that, you generate a lot more data, of course. The data can automatically transfer to an ELN. People can access the data from their home or wherever they're quarantining and can do the analysis very quickly and instantaneously and can deploy potentially the next experiment. On the clinical side, telemedicine became a reality almost overnight. It was at least as a patient myself sometimes or with a family, I was not aware that any of the physicians that we were interacting with were using telemedicine, and suddenly I was getting emails and phone calls saying, you know, we've got telemedicine if you're interested. Even physical therapy appointments became available via telemedicine, which was also quite interesting. Direct to patient became much more of an attractive and feasible thing, I will say. Being able to, during this global pandemic, when the patients either can't or aren't excited about leaving their house to go to a hospital of all places, being able to still participate or continue to participate in a trial by having the drug being shipped to them, having the technology capabilities to be able to communicate with them and make sure that they're still safe and be able to monitor how they are doing is actually quite remarkable to see, and quite remarkable to sense the urgency with which people wanted to get on and start using the technology. And thankfully, we were down the path of digital transformation so that we had a lot of the technology available, partially deployed, to be able to deploy much faster in appropriate places. Obviously, I don't want to give the impression that all our clinical trials became, direct to patients, because that's not really possible with all of them from where we were. But certainly, we could evaluate trial by trial and make informed decisions and have the technology available to say, okay, with this trial to these patients, we can actually continue the trial and not have to abort. With all of that in mind, and if we look at the future, kind of being a little bit more of the crystal ball, what can we see coming in the future? Certainly harnessing AI and machine learning and to improve R&D efficiency and productivity is key and is what we're driving. I've got the picture of a robot. We're not too far from that, actually. Robots who are able to monitor how experiments are going, how the robotics processes are progressing, can replenish reagents, automatically keep track of reagent consumption and replenish reagents, multiplexing instruments. I think we're moving in the direction of printable lab consumables in some areas. So there's a lot of possibilities and options. Will the labs ever become completely without the expert scientists? Absolutely not. You always need the scientists. But certainly we can make better use of scientists' brains, if you will, and have some of the manual work be taken up by automation. When it comes to clinical studies, and I noticed I'm hiding the tomorrow there, clinical studies of tomorrow, telemedicine clearly is here to stay. Siteless clinical trials are becoming the norm, and it's the right thing to do because you can reach more patients. Patients who can't move to get places, can also participate in trials. External control arms are also becoming more routine, or virtual trials, not yet, but I think certainly to be able to feed the design, being able to run a trial virtually first with digital twins of the patients in order to feed better design is absolutely key, and that is in the future. Electronic real-time monitoring of patients, the drug response, safety, efficacy, patient-reported outcomes, getting that in real time and not with the delay that it takes to wait until a patient goes to see the practitioner in order to report the outcome and so on. Proactive alerts to healthcare providers and to patients is also fairly common now, but it'll accelerate even further. And finally, access to, for the patients to have access to their own data is much more prevalent now and will continue to be more prevalent, enabling the patient to be more empowered and more in control of their therapy. I put a statement at the bottom there. Kind of my vision actually is no experiment, study, or trial is designed before we do an evaluation in silico. Again, making use of all the data that we've generated over time in order to make the best possible decisions and the best trial. My final slide here is really just to highlight on the path here of what's coming in the future. Some of this is already here and now. Some of it is, looking into the future, actually. like VR portfolio modeling, flexible partner network, and so on. Paperless Lab is something that we're working towards. These are all things we're working towards at various levels of maturity across the pipeline, and it's certainly not a complete list. I will end with, what does an R&D scientist workbench looks like? Well, we're used to having and want to always have things on in our hands, right? So having them on our favorite phone, having lots of gadgets from which we generate data. Alexa, get me this reagent or that reagent. Having that voice recognition and being able to use robotics to get work done more effectively, more efficiently. And some of you may be thinking your desk at home already is looking like this, but effectively the desktop of an existing scientist, and certainly in the future, will look more like something like this. You've got your laptop, your gadgets, your sensors, as well as your hopefully not as many handheld devices. Right now, we still sometimes have to use more than one handheld device, but effectively, you're able to get a lot of the work done from your desktop and being more productive and more efficient, and having much better data and information for decision making. And with that, I will pass it on to Oliver, I think. Yes?
Thank you, Anastasia. Really appreciate your remarks. Fantastic vision for the future. There were several questions that came in while you were speaking. I'm going to take one of them now, and then I'll save the others for the panel discussion. And then we'll introduce Oliver. So let's see, how can I decide? I think I'll take the last one that came in. The increase in digitization and automation in clinical trials, that this will it eliminate phase 0 studies in animals?
Will it eliminate phase 0 studies in animals? Only when we figure out how to translate preclinical data to clinical outcomes. So is the goal to reduce the number? Yes, absolutely. Can we completely eliminate it? I probably need a toxicologist to weigh into that a little bit more. I would tend to say reduce the number. Yes, reduce the usage of animals. We've been working towards doing that. Would you be able to reduce it to 0? From where I'm sitting right now and the information that I have, I think not for a long time. And I think a toxicologist would probably be able to weigh in on that a little bit more. But certainly reduce the number, yes, for sure. Reduce the number of animal studies that we need to do. But a prerequisite is for us to master that translation, if you will, between preclinical observations and clinical outcomes for existing data that we have. The more we learn about that, the better predictions we can make, the less animal consumption and preclinical testing we will need.
Terrific. Thank you so much. There are more questions to come in a bit. I'd like to turn to Oliver Hesse, who is the head of biotech data science and digitization at Bayer. Brief introduction of Oliver. His career started in small biotechs, building up and running high throughput screening systems for large molecules that generated large amounts of data, and incorporated in those roles automated decision making. He then took a role in 2008 at Bayer and has been working in Bayer Berkeley since 2011, where he's been developing and implementing strategies and projects with respect to lab automation, lab informatics, and data sciences. In a new role as head of global biological development, he's focused on the application of data science and digital to gain better process understanding, plus digital integration from development all the way to manufacturing. Today he'll be speaking on the digital continuum from development to manufacturing. And thank you, Oliver. It's all yours.
All right. Hello, welcome. Good morning, good afternoon, wherever you are. I hope you can hear me, can see my screen. Tim, maybe a short confirmation?
Yep, all clear.
All clear, great. So as Tim mentioned, what I'm going to talk about is a little bit around the continuum from development to manufacturing. I will talk not about the latest and greatest technology, honestly, but more about some basic concepts that we can address. However, while my talk is about technology, I have to bring this disclaimer first, right? These are forward-looking statements, and certainly also this is not an endorsement or recommendation for, or I'm not providing any endorsement or recommendation here. Now, before I start with any kind of technology, I want to start actually with the most important aspect really around digitalization or any digital concept. And the main or the most important topic is really culture. As I say here, the main challenge isn't technology. It's really people. So while the technology is basically there, and you will see some of the concepts that I'm describing are fairly simple. But even more complex technology, it's there, it's proven. I'd say other industry are adopting it in a much higher rate. We see it also on consumer side, and we saw great examples from Anastasia around technology that we're using in our daily life, in our daily home. However, if you look at the workplace, it looks somehow different, right? And really, in the end, it's about adoption, it's about culture, it's about how do people actually adopt to those new ways of working, or how are they embracing it. And in the end, it's around a mindset question. So am I willing to share my data? Am I willing to give up maybe some grab that I have on the data? And it is certainly also a big aspect of data literacy. So how educated is your personnel? How educated are your employees around what data can actually provide and what digitalization means? We heard it also earlier, what does actually digital transformation mean? There is better or worse descriptions around it, but it's a vague concept, right? So this is really something that's at the heart of any kind of digital project, of any kind of digital transformation. It is really people. And I pointed out a few things here, right, that really transform. So it is really about the change from siloed work to more interdisciplinary collaboration. It is about data-driven decision making, and really at the front line. That means people need to have that data available, right? It's around data democratization. So every data point is available to every person, and so that they can make those data-driven decisions and not waiting for somebody else to provide their data to them at a point of their convenience, right? And it's certainly also a different mindset in terms of being much more agile, being much more experimental and adaptable. And I really like what Anastasia presented a little bit earlier, that before we do an experiment, we actually do an in silico experiment, or we run a simulation. Because that is something that can speed up things significantly and make the work that we do in the lab as a wet lab experiment much more meaningful. I also provide here a little link to an interesting article in the Harvard Business Review around the AI-powered organization, where they really come to this conclusion here around its culture and not technology. All right. Nonetheless, we want to talk about technology as well. And when I talk about the digital continuum, I'm going to zap through this little animation here. I start off with this picture also internally quite a bit to explain a few of the concepts. And it's a busy picture, but if we start from the bottom left, and we're looking at what are we thinking about, what is the digital continuum anyways? So we're starting really in development, process development, maybe multiple sites, Then we have clinical production where we need to go to, and then we have our manufacturing. And ideally, in this digital continuum, what we have is a certain alignment. So that we don't have many, many different automation systems, all kinds of different physical equipment that goes around there, but we have certain alignment here. There is something then that is really, really important in this continuum is the concept and idea of parameterization and modularity. We don't want to reinvent the wheel all the time again. It's not necessarily in the best interest to of the process development and the whole process to be creative on defining what a chromatography is, for example. Right? It is very important to vary and be creative on the inputs and outputs. However, to define what a chromatography is not necessarily the most valuable aspect here. So that's an important piece. The concept of ISO S88 gives us some guidance around this as well. It is something that is used in many industries. From my experience in the pharma industries, there's some adopters. If you look at biotech in general, the concepts are not that widely, or that concept is not that widely spread. However, it gives us a good framework, an idea, and concepts that we can use. Now, very important then for actually the person working in process development or in the manufacturing or somewhere in between, we also need to have applications that support this whole idea. And the main message here, if you see it, look at this little application landscape, you can certainly discuss around the one or the other thing. But there's a couple of important points, and that is, or the most important is actually the concept of APIs here. What I say is basically those things need to be interconnected. We don't want to have data silos, right? We don't want to have individual applications where some data sits and nobody can get to it except for that one person that we knew 2 years ago that worked in department XYZ. So really having APIs, having the ability to query that data, to interchange it, or to interface with different systems is very important here. Now, there are certainly more aspects to it. I use this picture also quite a bit internally. There is another complete layer on top of it that I'm not going to touch upon today, where we think about the application of data science, AI, machine learning, and so on and so on. I'm not going to go into that today because I really want to focus on one aspect here. So, I like simple pictures, and I try to make it as simple as possible here. What does it show you? Basically, as we said earlier, right? As I said earlier, our endpoint to some extent is manufacturing. We want to bring a process into manufacturing, and this is really the goal of process development. Now, if we start on the right side here, we want to get to manufacturing. Then we have a couple of other aspects that we need to consider. And that is really here recipe management. So if we think about our process that we want to use to develop or to produce our drug, our biologics, or any other drug for that matter, we can describe it in a recipe. But however, then the recipes need to be managed, right? So we need to have this recipe management. Now, then there's a couple of other aspects, and that is we want to actually develop a process, and we're going to go through iterations of a process. So we need the idea and the concept of a process management. Specifically true if you think about distributed work, where many people work on that process. Then we need to have versions and iterations, and I think Tim earlier alluded to that as well. What are certain branches of a process, right? How do we improve that process? So we need to have a concept around that. There's another aspect of risk management that fits together with this. So we need to develop control strategies. We need to have an assessment of the risk around this process. So that needs to be managed as well, because also there, over time, we're getting more insights. We may develop this further. And then honestly, one of the important pieces, or most important pieces, actually, we need to talk about reference data. We need to have something that glues all this together. And we need to have, of course, you see all the arrows back and forth, interactivity between those concepts and ideas. Now, this is actually my main slide. And what I want to do is I walk you through this. It is quite a bit in there. I have a version that has much more on it, but I tried to simplify to really get to the major points here that I want to talk about today. And let's start with the reference data as I mentioned it. So really the main point here, and it's really the basis for everything, is that we have a good idea of what are we actually talking, what language are we talking. So the idea here is we need to speak the same language. We need to have a concept of what it is that we are describing. And that can, I mean, there's concepts around this along taxonomies. And then if you take that a little further towards ontologies, right, you can get there really, really fancy with semantic technologies, and it can become quite complicated. But those are ideas and technologies that are proven and out there. And let me give you a quick example of why this is important reference data. And specifically, if you talk about also distributed work and many different sites or many different groups working on something together, right? Or if you have data in multiple systems, and you want to really make sure that this is the same, and we're talking about the same thing. And it could be something as simple as some alignment on certain topics. So conventions, for example, what is actually day zero? Is it day zero at a cell culture, or is it day one? So a very simple idea, but it needs to have some alignment. And we need to have that also somewhat ideally programmatically represented, and not only on a piece of paper where people can adhere to, or maybe not. Another idea is talking about a flow rate. What is really a flow rate? Or we can speak of simple things like the actual nomenclature. Is it cell viability, or is it viability of a cell, or is it cell underscore viability? So those things need to be aligned. And again, very important specifically also, if you talk about distributed work, and also if you work in different settings, maybe also if you think about the current situation, many people working from home, that you have ideally something that informs the other systems what nomenclature and what language to use. Now, if we move forward to the next part here, that is around the process management. So and control strategy. So we're developing the process usually in a very collaborative way. So there's multiple groups, could be all over the continent. And I think the paradigm in the past was usually, hey, I'm looking at experiments. So we usually have a very experiment-centric view on this. However, in the end, as it says, right, we want to develop a process. So we need to have also a concept of what does a process actually, or how is the process represented? And what is how does it manifest itself? Is it a piece of paper where I describe a process, or can we programmatically represent this process? And that is, again, specifically important if you work with multiple people on this, multiple groups, right, where you can have individual experiments, but you need to bring them into the context of the overall process that I'm trying to develop. And I have this little idea here about the templates. I recently saw a presentation, it's already 10 years old, that introduced the concept of reusable manufacturing actions that I like very much, right? Again, goes back to the point, I don't need to reinvent what a chromatography constitutes, but I want to use this concept and reuse that to build my processes and to bring variations then to the actual process parameters. Similar in that regard, control strategy, getting from my target product profile to the actual control strategy. Again, here, distributed if we have the capability to programmatically represent this control strategy and also the way to get there. So our risk assessments, ideally build them on templates. Multiple groups can work on them simultaneously. And also over time, right? We're learning about this over time. We may readdress our risks. So that's something where we need to have the capability to, again, represent this programmatically. Now then, really, as I show here, those things should interact with each other. And then what we can do is we can hand over our process that we have developed iteratively over time, phase 1, maybe phase 2, phase 3, whatever it is. It can hand that over to basically our recipe management. And I'm going to talk about that for a little bit. So the recipe management here really is a description of our process, but in an abstract form. So if we think about kind of the analog world, how is it usually done? Well, process scientist develops this process, her process. They have some structured data around experiments, right? Then they write a relatively wordy process description. People sit together then with maybe the manufacturing science or the operations group, and then discuss basically the individual parameters that are more or less described. And then we go and implement in the manufacturing areas, be it clinical or be it really commercial or launch facilities, right? However, that is pretty much a deviation. You have the underlying, if you have the underlying data in a structured form that really constitutes the process, and you have a programmatic representation of your process, it is possible to actually hand over this process from machine to machine. So that doesn't mean that you eliminate the scientist in it, but it allows you to circumvent some of the interpretation that is currently going on, or that can be going on if you have basically a written report. Somebody mentioned it before also, I think, or I saw it on one of the slides around regulatory expectations, right? Also the FDA in the end has at least the CASA initiative. And I think the movement is going towards we need to have the data in structured ways that we can actually submit them also in electronic form. So that's basically the idea here around the recipe management. And you see here, you can hand over this process into your MES system. From there on, pass it down to the DCS, driven by templates. And ideally, you can do that also, or you can feedback, does my process actually fit into that facility? And do that in silico instead of going in and then seeing, this process doesn't really fit because I need too much buffer or my bioreactor can't handle this, right? So it's really the digital transfer of those recipes. And here again, I think very important, and that's more or less my last sentence also, in the current situation where we have a pandemic, where travel across the world is really, really restricted, and companies certainly don't want their personnel to travel, how do you actually do a process transfer if you can't send the people around, right? So are there ways to do this, right? If we have this digital transfer of those recipes, that probably doesn't eliminate completely that you need some people, but it allows us to at least have the basic information really digitally transferred in machine readable ways so that we can reduce at least the need for any personal travel. And with that, I thank you very much and certainly open for questions.
Thank you, Oliver. Appreciate the stimulating talk. And we had a few questions come in. I'm going to give you one that came in, and then we'll bring Anastasia on screen, and I will come back through for a more wide open discussion and take some of the previous questions. So one of the questions that came in while you were talking was actually about culture. You mentioned how important culture is to transformation, and the question came up was, do you have any tips that can help encourage data and digitization perhaps faster? And I'm going to add to that question, is the impact of COVID and the work from home and the necessity of that making it harder or easier, the change culture.
So let me come to the last point. I think it makes it easier because a lot of people really see that paper doesn't work. If you're not on site, if you're not together in a group, well, the wet signature becomes a problem, right? So I think yes, the current situation with COVID and the pandemic, I think really drives digitalization, or at least it drives also the mindset that there is more concept needed. I have had just yesterday an email coming to me from somebody who is usually relatively reserved about those things. Well, don't you have a solution for me that is digital? So I think that is something certainly driving mindset here as well. Overall, I think it's a really good question. It's a very complicated question, or the answer is not easy. It's not straightforward. Traditionally, I would say, well, you need to show value. You need to show value for most people, right? What's in it for me is certainly a major point. Having that said, I think education and active management of change is really important. So really bringing people up to speed. What do many of those concepts mean? And really have educational series. I mentioned data literacy, right? So people need to have an understanding of what many of those things actually mean. Right? And I see that every day again when I also internally describe some of the ideas that you need to educate, right? It starts with simple things that we talk. What does digitalization mean? What is digitization? So really actively going out and over again, bringing that message and educating people of what things mean.
Awesome, thank you. Actually, I'm going to flip on my camera, and Anastasia, if you wanted, and Oliver, if you want to go on camera, so we have some semblance of a panel discussion, that would be great. So I appreciate the comment on education, and this has come up multiple times in discussions with people for a long time, and particularly recently. I'm curious, how is Bayer and how is Janssen approaching the education? Is there a determined and dedicated effort to creating time and space for people to learn these data science and digital tools, or do you approach it some other way? And that's a question for both of you. I guess we can start with Oliver since you brought it up.
Well, I don't know that we specifically carve out time for it. And to your point around data science, I think I see data science as a scientific discipline where you should have an idea of what it is, but it's actually nothing that you can learn just on a weekend, right? However, yes, we have programs. We're giving people the opportunity to learn by themselves. So we've actually partnered also with some online institutions to do that. We have, honestly, a load of information that sits more or less passively, of course, on our SharePoints and websites. And what we're also building is digital communities. So where we really try to identify digital leads that go out then and also actively then manage on a site or department level and educate.
Are you seeing it have an impact yet, or is it working? Would you change anything to make it more effective?
I think it still needs much more active management and really having the understanding of the role of also what a change management does, right? So I think there's still room for improvement, but I guess there would be always room for improvement, right?
And I'll take the question to Anastasia, and I'm going to spin it a little bit because you mentioned, and I'm still a little bit in disbelief on this, that your labs are actually running more or less fully automated. Well, not fully, but you're doing remote experimentation. And so it's a hybrid question. Is that for real? Are you actually operating experiments from your couch, and or is that more of a vision? And secondly, if you are doing that, how are you educating people? Because I'm sure there's a whole bunch of new things people have to learn to make that reality.
Right. So it is a vision and it is a journey. So we're absolutely not running all of our labs virtually. But what we do have is a lot of robotics and automation so that you can start the process and monitor it from home. And if it has gotten stuck or something, you can restart it from home. In a couple of labs, there's actually the roving robot, if you will, that I mentioned that someone put together just during the pandemic, just the ability, because sometimes your robot gets stuck and you don't really know. You can restart it, but you're not really sure what's happening. You can't see what's happening. So being able to have a set of eyes, if you will, there through a robot, a beam or something like that, that can go to the site of where you think, well, where the robot is, where the actual robotics engine is, and be able to see, through a video feed, potentially what's going on. That's a one-off that is being done in one experiment that someone tested it, and I think that's the starting point for us to be able to do more of it. But the idea of people go in the lab and they get experiments started, as long as you have the robotics set up, you can set it up, then you can go back home. You don't have to sit there and babysit it and watch it, and you can monitor it from home. That is available, and I don't think it's uncommon elsewhere as well. But we're absolutely not to the point of being able to work completely outside of the labs. Will we be able to get there 100%? No, I don't think so. We're not eliminating the need for the scientists, but having the scientists move to higher value activities actually is what we're driving towards.
So how do you approach that? That's a training issue, and more than training, that's a deep educational issue of spending much of your career learning how to operate physical equipment, to learning how to move bits and bytes around to do deep data analytics that really was only relatively recently has become possible. How do you help your staff make that transition?
Yeah, so first of all, it's a collaborative effort. Secondly, we've had our head of R&D has very clearly stated that he expects all his scientists to be bilingual in both science, whatever their science is, and data science. So, and then it's the collaboration, right? They're not going to suddenly be machine learning experts, but they're going to know when they need to employ an expert for machine learning. They're going to be more open to, as part of the change management, right? More open to different ways of doing things, doing more predictive capabilities, generating lab, using legacy data actually to inform your design of your experiment, so that the question earlier, you can reduce the number of animals that you might use, or you might reduce the number of iterations of the experiment that you're doing. You might design your experiment more effectively. So it really is a collaboration between the data science expert, who is more data-driven than science-driven, but still can do both, and your scientific expert, who understands the data science, knows when to, what language to use and when to bring in the expert data scientist, and also is willing to try new ways of working and take the prediction results and put them to the test in the lab.
Fantastic. And on that subject of more predictive and more efficient experimental design, one of the questions from the audience was the use of digital twins. And I realize that digital twin is almost, it's a wonderful word, because or two words, because you can essentially imagine it to be anything you want it to be. But I think that the thrust of this is, could digital twins or simulation or automation allow you to predict experiments to reduce the need for actual experiments? Will we get to a predictive capacity of that type of fidelity, almost like the way you can simulate airflow across an airplane and not need to run the actual test flight?
I think
right. Before, oh, go ahead, Oliver.
No, just one thought there, because you mentioned can we limit experiments, right? I would see it in different ways. It's a different type of experiment, right? I think you can distinguish between like the wet lab experiment, where you do some would say the real experiment, right, in real world, versus an experiment in silico, right, where you use maybe predictive models, and that could be data-driven models. It could be also mechanistic models or some hybrids around it, right? So whatever you know mechanistically, you should not throw away, right? But can you augment it through data-driven approaches or vice versa, right? I think that's where it goes, and that's also the idea. I would argue for the digital twin, right?
Yeah, I would also say that it's, so I agree with you, and I would also add that it is it's a journey, and you're taking steps towards, in some cases, you might not need to do an experiment. In other cases, you're designing a better experiment with less repetitions, with less animals, with less samples, less patients, if it's a trial design. And that's really the starting point. If we can routinely do that, we're accelerating drug discovery development significantly. So you don't have to reduce it to zero in order to accelerate things. But part of the digital twin, and I think we have examples in the pharmaceutical development, so bordering on manufacturing actually, where you can use legacy data from different recipes that have been scaled. You can use that data to actually inform a new scale-up process that you're trying to develop, and that there are examples actually where that's been used effectively.
Yeah. One of my favorite analogies, and people very often, it goes back to educating people, ask me for analogies, right? What does it actually mean? And one of my favorite ones is actually car racing, Formula One, where they have very little time to actually prepare. They have very little rounds on the racetrack that they can actually do, right? Nevertheless, they need to prepare their car. And what they do is they have relatively sophisticated models that are mechanistic, that are data-driven, all kinds of what they know about their car. They put it in, look at the track, look at historical weather patterns, I don't know what, and they do simulations, right? And to find their first, or the optimal setup for the training round. And then they just go on the training round and basically confirm their setup, right? And that's kind of how I would envision it as well, right? So you can do simulations, you have your optimal setup, as you said, in some instances you may not even need then the actual experiment, but you can do much better, much more informed experiments and much more meaningful.
Terrific. So we're almost out of time, and I'm going to give each of you one last word. Oliver first, and then Anastasia, you get the last word. So the simple question in a minute or less is, if you could change one thing right now and snap your fingers and it's done and it's implemented, what would you pick first to advance your digital transformation efforts?
I think standardize, having everything standardized, terms, interfaces, connectivity, would be a huge benefit overall, because that's a lot of what is holding us back next to the cultural aspects, right? But standardization is something that where we don't need creativity, we want to have standards, and that's a big thing. It takes a long time, and if we can do it like this, would be great.
Perfect, thanks. And Anastasia?
I'm going to take Oliver's from earlier, culture. I think that's one that I didn't mention in my talk, but culture is a huge one, and there's different aspects to that. It's the change management, the willingness to do things differently. It's the acceptance that just because we're doing things differently doesn't mean your job is obsolete. You're moving on to other bigger, better things that you're doing, and being more accepting, understanding that different perspective and accepting of a new way of working and trying it out.
Perfect. So for those who don't want to listen to the whole video, we can boil it down to three things: standards, culture, and education. And that's it. We're done.
Yep.
Awesome.
What's there?
The age old,
Right.
So thank you so much for your time this morning. I really appreciate it. And thanks to the audience as well. Sorry we couldn't address all of your questions today, but I did really enjoy it and appreciate your time. And there will be a recording of this posted at some point in coming days on the BioIT website, and links to that from Riffen's own website.
Fantastic. Thank you, and to the organizers too.
Thank you, everyone, also for listening.
Okay, thanks.
Okay.
Take care.